All right. Okay, so we're going to start right now. We have a sizable number online, so we're not going to delay this. So I just wanted to welcome everyone. So this is part of like our program called Discovery Spatial, where we would like to introduce different platforms to our community here. So we know that there's been several platform developments lately, and we have a different variety right now that's showing up. So the more we know what are the different platforms out there, I think the better we can be in terms of designing our spatial experiments and how we can combine them in interesting ways. So that's the whole, what do you call it, the concept behind spatial discovery. So starting this off, we are going to introduce Stereo-seq. These events will be recorded, so they will be online if you cannot attend today. So without further ado, I would like to invite our first speaker, Yun Zhang, who's the head of Stomix. Now, I'm gonna see what's wrong with this. Okay. Yeah, you can start.
All right, hello everyone. Thank you for joining me today. So my name is Yuan. I'm the head of Stereo-seq. Today, I want to share with you a groundbreaking technology called Stereo-seq and how it can accelerate multi-omics study. Let me see if I can use the laser here. Give me 1 second.
So the laser pointer probably will not work. Yeah, so I think you'll just have to use the mouse pointer.
Are you able to see the mouse?
Yeah, they will be able to see the mouse.
Okay, then I'll just use the mouse.
Yeah.
Thank you.
Can they see the mouse?
Okay.
Yeah, they can.
All right.
Okay.
So before I dive into the specifics of our technology, I'd like us to take a quick look at the journey of medicine or a treatment, which involves a lot of critical steps illustrated here that ultimately reach to patient's bedside. And then this, so historically, medicine discoveries is using a one-fits-all strategy. While it works for many, it doesn't take into account of the unique factors that affect individuals to respond to a specific medicine or a treatment. So that's where, the precision medicine comes in, optimizing the health outcome by leveraging the advances in technologies over recent years. And the technologies shown here on the left bottom figure. So it has provided a comprehensive tool set for researchers for precision related discovery, such as now we can do whole genome transcript, WGS, whole genome sequencing, RNA-seq, and targeted protein panels. And we can also look at epigenomics and metabolics, like different aspects of a variety of sample types. Translational studies normally involve patient samples, animal models, or even organoids. And a lot of assays we are talking about here, they are just doing or using bulk analysis, average, an assay for average expressions. However, it doesn't provide us very critical in situ informations. And the spatial information is really critical for us to understand a lot of biologic questions. Shown here, as you can see in the middle part, this is the bulk RNA analysis, like I just mentioned. It assay for average gene expressions from mRNAs of given tissues cells called transcriptome. And then single cell sequencing technology was developed, and it can add brings a more fine-grained assessment of each cell's transcriptome. And then spatial single-cell transcriptomics even going one step further, that can really provide or maps gene expression back to its original location and provide positional context of those gene expressions. If we're focusing on the single cell transcriptomics for a minute, it's primarily grouped into two categories. One is image-based. The other is called sequencing-based, mainly NGS, next generation sequence-based. And then for the imaging-based, a lot of those techniques, they are using in situ hybridization, which gives us higher sensitivity and it can reach subcellular resolution. However, it requires prior knowledge to design those detection probes, and it's tentatively no throughput, because when you do the assay, when you design the panels, the size ranges from a dozens to a few thousand. On the other hand, sequencing-based technology, they can capture the entire whole transcriptomics, and it's very high throughput. However, before Stereo-seq, the resolution and sensitivity is relatively low. So and then so back in 2020, last year, so 2020 is single cell transcriptomics was selected as a method of year in 2020 by Nature method, and last year single cell proteomics was selected as the method of the year. So which also gave us more insights on the average expression of multi-omic data. And this is a very key component to for us to understand the biology of the health and the disease, because it can help us to create this Atlas project, which is an effort to really map and characterize every single cell type within a given tissue, and that will help us to that will help researchers to understand the, for example, cell-cell interaction and cell dynamics during normal development stage or disease stages. So if we're going back to the NGS based spatial tools and look at how underhood biochemistry works for those NGS-based spatial tools, there are two main strategies. One is using poly-T probe, so that poly-T will allele to poly-A-tailed RNA and capture the poly-T adenylated RNA, which is normally mRNA. And this is very helpful because it will help us to get the 3' expression of the RNA profiling. But it's still missing information. For example, it doesn't capture long-coding RNA or some of the microbiome RNA, which do not have poly-A tails. So therefore it's it's still limited in terms of as a discovery tool. And on the other hand, there's probe-based method, for example, 10x Visium platform. They are using this probe-based method. So they designed, probes to capture, for example, up to 19,000 genes, which they call whole transcriptomics. And then that will provide some insight to the genes of interest, because it requires prior knowledge again to design those probes. Therefore, it is still not the best discovery tool for anything if you want to capture anything unbiasedly. That's why we set out to develop a better spatial discovery tool with three main factors into account. One is throughput. We want to still maintain high throughput that we can capture whole transcriptomics unbiasedly, and we also want this tool to be species agnostic, so people from any field or any application have any application can use this tool. And another important feature we want to have for this technology is that we want to reach single cell resolution. If you look at the average cell size from this chart, it ranges from less than smaller than 1 micron to slightly bigger than 10 micron. So that's the average cell size. So that's why we want our technology is targeting to reach a resolution smaller than 1 micron, so that we can really reach single cell resolution. See why? Oh, okay. All right. So our technology is called the Stereo-seq, spatial enhanced resolution omics sequencing, and this is developed by Stereo-omics and company Genomics. This company is an exclusive distributor in the Northern American region of Stereo-omics. So this takes advantages of a proprietary technology called DNB technology. So we are utilize this sequencing chips and directly apply the them to capture the tissue or RNAs from tissue sections. And this in silicon chip is a patent array. And on this chip, there's small dots we call DNB spots, which is only 200 nanometer in diameter. And then the center to center distance between spots is only 500 nanometer. So that is very, very densely packed chip arrays for capture. If you take a look at the upper image here, so that is a section mounted on our chip, and we did a nuclei staining on this tissue section, and you can see this is a single nuclei, and then the dark black spots are the DNB spot, which is our coated with the capturing probes. As you can see here, within a single nuclei, we have hundreds of those capturing spots in this nuclei. So that really help us to reach the resolution at even subcellular level. And on each individual DNB spot, it is coated with hundreds to thousands of capturing probes. And those capturing probes from the same spot, they share the same CID barcode, which is a positional barcode. So this positional barcode will help us to map the captured MRI back to its original location. So that serves as sort of like the X, Y, axis grid. And then each probe from this capturing spot, DNB spot, has a unique UMI, which is a unique molecule identifier, which will help us to count the RNA transcripts, the number of RNA transcripts we captured. And there are two designs of our capturing probes. One is we're using poly-T probe, just like what I mentioned. It can capture total mRNA. And then we have a second design. This is unique from any other technology from the market. This is a randomer designed, so it can capture total RNA, including non-coding RNA or any RNA from microorganisms in the tissues. So after we use after the chip is manufactured, we can mount the tissue section onto our chip, do the in situ RNA capture and cDNA synthesize, make the libraries and sequence them, and then we have also in-house developed algorithm that can help us to map the RNA transcript back into individual DNB spot, which is only 200 nanometer, 200 nanometer in diameter. So that is very, so it is very dense and the resolution is high, as only 500 nanometer. And again, because of the design of the capture probe being poly T or random N, we don't need prior knowledge for those regions, so it can capture everything. It is also species not agnostic, so while capturing total mRNA if we're using poly T probe design, or total RNA if we are using randomer N design. And our technology also supports different staining methods on the same section. As I just mentioned, that we can take the nuclei image and then we can use this nuclei image to do some imputation when we had in the downstream analysis. And then we can outline the putative cell boundary using the nuclei image and mapping the transcripts back to individual cells, like showing here. This is using cell beam for the data analysis. We can also support H&E staining. So this can provide more pathology information and gives a more accurate region-specific transcriptomics data as shown here. Oops. Oh, for some reason, this doesn't support the animation. Yeah. Anyhow, I was trying to show you the antibody can also be incorporated into the process, and there are two protein stainings on this mouse testis, CD4 and
Yeah, it's not showing the embedding.
yeah, because that should be pop up after the slides. Give us 1 second to see if we can fix the animation problem, or can we move this around to show, but not this one, the entire group.
Oh, this entire thing.
Yeah, the entire group. Let me just, if we cannot use animation, let me see if I delete it. Okay, yeah, I'll add it back.
Okay.
So in this slide that we added antibody staining before capturing RNA, and then we used antibody CD45R and CD4 to do the staining. You can just use commercially available antibodies. And then the mouse testis RNA transcriptomics information can be coupled and co-localized with the antibody staining as shown here. You can see, you can get a full picture of protein information as well as RNA information, providing multi-omics information. Let's see if I can bring what we had just now to do. Okay, yeah, that's how animation works now. So anyhow, just to repeat what I told you, that we can do nuclei staining, and we have a primary analysis pipeline that will help you to map the genes back to individual cells, achieving true single cell segmentation. In addition to nuclei staining, we can also do membrane protein staining that will provide more accurate single cell segmentation. And then we can also do H&E staining or the antibody staining, which will help to provide multi-omics information. Okay, don't highlight anything here. Go to next page. Let's see. Sorry. Okay, should work now. All right, so in addition to analysis pipeline, we also develop what we call Stereo-seq This is a in-house developed visualization tool. As you can see here, this is very user-friendly. You can do spatial single-cell data analysis by selecting, for example, different regions and then so out the region of interest and looking at spatial distribution of different cell types or cell clusters. And this is also supported by any third-party single-cell segmentation algorithms. Another very unique advantage of our technology, which is not provided by any other company, is the our field of view, or we call it chip size, is customizable. Because we're directly utilizing the sequencing chips, and our largest sequencing chip for now is 13 centimeter by 13 centimeter, which is definitely not the upper limit of our technology. However, for for the commercially available products, we have 0.5 centimeter by 0.5 centimeter all the way to 2 centimeter by 3 centimeter. As you can see here, you can fit a mouse brain embryo or a mouse brain on this 2 centimeter by 3 centimeter nicely, and there are different customizable chip sizes. For example, there's 3 centimeter by 5 centimeter, and this 6 centimeter by 6 centimeter. This can fit in the entire macaque brain. So the customizable sizes of our chip design really fit different organ or tissue needs for different research projects. So again, I just want to highlight our advantages of our technology. I just mentioned, I went over a technology that can provide true single cell segmentation at single cell resolution, and we have large chip design, which can also provide multi-omics information. So this powerful tool can really help us answer a lot of questions from different application fields, such as neuroscience, oncology, aging, evolution, or development, etc. So in the next few slides, I'm gonna give you a few examples of trying to answer questions from different application field. So this is the world's first 3D single cell atlas of the macaque cortex. Researchers have been using this large chip design, 3 centimeter by 5 centimeter. They were using more than hundreds of those large chips from 161. They also used the 1 by 1 smaller chip design. So total section they were doing for this study is 161 sections from 3 macaque brains, and more than 50 million cortex cells are spatially mapped. And then the macaque cortical cell taxonomy was established using the 264 identified cell types from this research works. And then they also discover that regional density and composition of cell types by analyzing the cell-cell interaction and cell composition, and that they found it is coupled with cortical hierarchy. And in these research studies, because our technology is species agnostic, so they also applied this similar strategy onto different species, and the cross-species analysis revealed a primate-specific cell types enriched in this layer called layer 4, and this really provides further insight on understanding the development, evolution, and aging, as well as the pathogenesis of the primate brains. This is a very cool paper published as science cover paper back in 2022. So researchers in this study, what they did is that they look at the axonal telencephalon development compared with its normal development process versus a regeneration study of 60 days. So this time course is 60 days. So they damaged half of the brain, and then they compared the damaged brain to the control brain within a time course of 60 days, and did Stereo-seq, the spatial analysis of the cell types and marker gene expression, and look at cell-cell interaction and cell dynamics. Interestingly, they found a subtype of glial cells actually was activated by the injury and then later compensated for all the lost neurons during this regeneration process. And through analyzing the cell-cell interaction and cell dynamics, they also discovered that this regeneration process actually partially recapitulated the normal development process. This is can never be discovered without a spatial tool like this. And our some of our US customers are using similar strategies to study liver regeneration, and their data should be published soon. So another application field is trying to do oncology. So dissecting and understanding the tumor ecosystem is essential to explore the mechanisms of tumor metastasis, and also trying to find a new treatment. So in this study, people combined the single cell sequencing data along with Stereo-seq, and they identified in the liver in patients with liver cancers. They identified this 500 micro zone. They call it a tumor invasive zone, bordering tumor site and peritumor site. And in this particular 500 microzone, they detected strong immunosuppression, metabolic reprogramming, and some severe damaged hepatocytes. And in a subgroup of those damaged hepatocytes, they actually found overexpression of two genes, serial amyloid A1 and A2, connectively called SAAs. And then in later this clinical association study, they used 5 cohorts from more than 400 patients of primary and secondary liver cancers. They found that patients with overexpression of those two SAAs in this tumor invasive zones in the hepatocytes actually had worse prognosis. And then they went one step further. They used the in vivo mice model. They knocked down those SAAs in hepatocytes, and they found decreased macrophage accumulation and delayed tumor growth as shown here. So this suggests a potential new treatment for liver cancer. And as I mentioned, this is a species agnostic tool, and a lot of people from plant field are also trying to apply this technology in their research studies. And this is a recently published, I think it just published 2 weeks ago, on a molecular plant. And this group used trying to create an integrated transcriptomic atlas of soybean during all their soybean organ development. So they have bulk RNA, so assay for average RNAs from 314 samples, and single nuclear RNA-seq and Stereo-seq from 5 representative organs, including root tip, shoot, apical, leaf, stem, and root nodule. The size for single cell sequencing and stereo-seq or spatial sequencing is significantly smaller than bulk RNA because the for now the price is still a little bit more costly for those assays. And then in this study, they found that using the differentiated expressed genes, they find that there AP2 transcript factors such as GMP PLTs, they play a very important role maintaining the stem cells in soybean. What's more important is that this HD-ZIP1 subfamily genes, those they found two group of those subfamily genes. One is functioning at the brown module, and the other one is functioning at the meta module, and they are regulates module development through different mechanism by targeting different genes from different cell clusters. This can never be discovered if without any spatial information. So this is a main take highlights from this publication. And what I just mentioned, those few examples are using the poly-T probe designed chip. So it's capturing the total mRNA. As I just mentioned, that we also have another design of a chip, which is a poly-N or random N design can capture total RNA, and this is compatible with FAP sample plot. So if we take a look at what additional benefits this randomer priming can offer, as you can see here, this is the whole gene body, and there are different technologies, including our, for example, the light blue one here is the poly-T designed Stereo-seq, and those are capturing 3' RNA information quite well, but only our randomer design can capture the whole gene body unevenly. And also when you compare with targeted solutions, in addition to our technology is labeled in blue, and in addition to capturing protein coding regions, we can capture a lot of non-coding RNAs. For example, non-long coding RNAs, small long coding RNA, and some microRNAs and some other RNAs. So it really provides a full picture of the RNA profiling. And in addition to the total RNA, it can also capture the microorganism or RNAs from microbials. For example, this is a study, again, time course study on mouse non-infected with TB. And we can use Stereo-seq to look at the spatial localization of the MTB after different different time points. And then when we look at its relationship with other, for example, immune response, we can see a negative correlation between MTB contents and the BCR related gene expressions during this time course study. This data is not published yet. And one of our US customers from MD Anderson, so they are using our technology creating this 3D integration of from different technologies. For example, they are using one section for histology by H&E to provide histology information by H&E staining, and they are also looking at lung-targeted metabolites using this technology called MSI, and targeted protein omics using a technology called COMET. And then they have one slice using our technology, which can provide whole transcriptomics information in addition to microbiome information. So they are using this 3D integration method And the panomics is also helping them to integrate those 3D information and trying to understand the tumor microenvironment in ovary cancer patients. And so this is not a complete list, but showing you some of the tissue types and organs being tested using our FAP workflow, and a lot of human and mouse tissue and organs. And there's also some non-human and non-mouse model systems have been validated using our technology. So just quick review of the Stereo-seq workflows for our poly-T design chips. And the wet lab process is about a day prior to make them to sequence on sequencer. And then for all FAP, that's around a 2-day process. And you can also add a 2 hour of RNA depletion toward the end of the process if you don't want the information of the RNAs. Yeah, of course.
The random time chip, how much of that is subverted to ribosomal RNA?
You mean how many the percentage of the RNA?
Yes, if you don't deplete, then it won't work.
It will work. It will still have, so typically it's about 20 to 30-ish RNA information we have from the data. Yeah. So not everything, with the majority is still mRNA and some of the non-coding RNA information we want. Yeah.
I guess I'm a bit surprised that you only lose 30% of your signal to ribosomal.
Yeah, probably because some of the probes are within the nucleus and that captures many the RNA information we want. Yeah, it's spatial localization, right? Yeah. Another thing I want to emphasize that even though we have commercially available 2-chip designs, but our platform is an open platform for innovation. And this group from Cornell, what they did is that they actually added one step of polyadenylation, adding poly-A spacing to total RNA on our chip before we capture RNA. Therefore, they can directly use our poly-T design chips to capture total RNA. And then they did some pilot study and on the fresh frozen intestine samples. This is the RNA, total RNA they captured, and they did the cell clustering and annotation and look at the spatial localization of those different cell types. And then they also look at the microbial RNAs they captured and look at the spatial localization of those microRNAs. So we welcome any collaborators or users to just use our technologies for different creative method development. So Complete Genomics is offering a tissue to data end-to-end solution. So here the whole process is that after Stereo-seq experiment, we have our DNB sequencing platform that can help to sequence the Stereo-seq libraries. And we also build our bioinformatic pack that can help you to do the primary analysis as well as visualize the data. So for the Stereo-seq kits part, what I just mentioned that we have the Stereo-seq transcriptomics, which is a poly T design, and the Omni random N design. And then we have large chip design, H&E staining and protocol for H&E staining, as well as adding antibody staining. What I didn't mention is that we also have a solution for high-plex of protein be captured on the same section. So you can buy commercially available protein panels with poly-A oligo conjugated antibodies, for example, from this Stereo-seq A panel from BioLegend. We validated on our pipeline and which can provide more than 100 protein information from the same section. So we call it Stereo-seq. And this is another solution that can help us to can offer multi-omics information. And we also have different a variety of sequencer choices for you. The T7 is the high throughput one that can help us to sequence Stereo-seq libraries at very cost effective price. It is about like $150 per whole genome sequencing. We also offer Go Optical, which is now a microscope designed for spatial technology. What it can offer is high resolution image as shown here, and then compare with other microscope. This microscope can really get large scanning field up to 10 centimeter by 7 centimeter at very quick time speed. It can finish scanning the largest area within 15 minutes, and smaller area like 1 centimeter by 1 centimeter, it can finish everything in a couple of minutes, which is very critical for any spatial related workflow, because you don't want the RNA to stay on the chip for too long, otherwise it's gonna get degraded. So that is the last slide of my talk, and thank you all for your attention. If you have more questions, maybe we can how to unmute, or maybe you can try to unmute yourself or type in the question in the chat box. Or you can go to those two websites. One is Complete Genomics for product information, and or Stereo-omics tech website for any technology question. Or you need more information, you can ask me here. Thank you for your attention. Is there any question?
We will take questions now. Let me just stop the share. Does anyone want to unmute and ask? Oh no, this is different. Okay. Actually, I have a question.
Sure.
Okay, I was curious about the microbiome. So how do you, so when I used to do GBM, when I used to do tissue culture,
Yeah.
there's always this issue
With
of contamination.
contamination, right?
Yeah.
Yeah.
So
yeah.
I was just wondering how do you actually take care of that? Like,
Yeah. So
yeah.
first of all, when you want to assay for microbiome information, you really have to have a very carefully designed protocol for that. And for example, you have to decontaminate everything you touch, and even for the experiment room, it has to be cleaned. And another thing is that within the design, you can also add the tissues uninfected, or if you, for example, if you care about the microbial information from intestine organs, maybe you can add a small piece, because our capture area is large, right? We can afford multiple mounting onto the chip, so you can have one intestine organs, and you can have a totally unrelated tissue from the same patient or model organisms onto the same chip to as internal biologic controls.
Do you recommend for those of us who are actually preparing tissues to actually follow a certain protocol? Because sometimes most of these like FFPs, like long-term storage, we can't do anything.
Yeah.
But can we? So we cannot do anything about it if
That's why maybe we can have some internal controls from different FFP like tissue types to make sure those and maybe have a few a duplicate for the samples to make sure the, what you see, is true to biology.
Yeah, but I think it's still gonna be difficult given the duplicates, yeah.
Yeah, but maybe, yeah, some other controls will help. But we did, so if you don't do things carefully, we did observe a fat finger sometimes, you know, from the microbiome information. When we did the first, microbiome test, yeah, actually, I have a lab at San Jose, we did have a fat finger onto those, when we, did the RNA profiling, yeah, which is caused definitely by environmental, not the, intrinsic derived from the tissue itself, yeah. Do you have a question?
So I have a question. Can this be used for strain identification in a metagenomic sample?
Strain identification, I
Like,
think.
for instance, if you have a tissue that's infected with multiple organisms,
Mhm.
right?
Yeah.
Is there enough resolution for you to I guess with the necessary barcoding, species identification, and all of it? Is it possible to do a metagenomic scan of an infected tissue and then tabulate the species of organism?
Yeah, that, I think depends on, the, how much microbial, RNA we can capture, right? And, for the infected ones, because you are, pumping a lot onto the, tissues, we are able to identify them. But if there's some rare strain or, you know, or low expressed RNA from those microorganisms, then, due to the capture efficiency, we might not be able to capture them at single cell level. But if we are able to group or make the bean size bigger, we might be able to observe them.
Okay, but in this case, we're not looking at metabolism. We want to illuminate each bacteria species or strain.
Yeah, look at their localization, right?
Well, presumably they would express something that's very high, that enable that strain to be
Detected, yeah.
So,
Yeah, should
so
be
can
able
it be used to, let's say, tabulate the strain in an infected tissue when there's like hundreds of species available?
Should be able to identify.
Has anybody tried it?
What I just showed you is the MTB infected mouse now, right? You can see at least one strain, and for other
No, but that's one strain. I'm talking about.
Yeah, hundreds.
Samples where there are actually hundreds of strains. Some are benign, some are pathogenic.
Yeah, we haven't tried that large scale yet, but based on the principle of the technology, I think we should still be able to detect most, if not all of them.
Yeah, because the field is definitely going to the fact that it's a community of bacteria that give rise to pathology, not because it's one pathogenic strain.
Agree, yes. Yeah. So really depend on the expression level, I think. If the hundreds or multiple strains have high expression, and we should be able to identify their spatial localization.
Yeah, just out of curiosity, just following up on that question.
Sure.
So the thing is, the number of strains that can be detected will be linked to the read counts and the dropout rate, right? The sparses field. So the thing is, I think that is kind of like the limiting factor.
Yeah, the cutoff of the to read for the readout. Yeah.
So I think they should be at a certain threshold before we can detect the strains, I think.
I think for the microbial RNA, our cutoff is relatively low, like 1 or 2 reads will count, this as identified microbial RNA. And going back to your question, I think our strategy is really a high throughput technology for high transcript sensitivity. So doesn't matter. For example, we can capture whole transcriptomics, right? That's 20,000 genes. So even you adding hundreds of microbial RNAs, as long as they have high expression, we should be, or relatively good expression, we should be able to capture them. Yeah, the limitation shouldn't be it shouldn't be limited by the number of microorganism or RNA transcripts. That's what I'm trying to say. Yeah. Does that make sense?
Yes,
Yeah.
and can you adopt this to look at DNA?
That's a good question. So there are some researchers thinking about, trying to do something using our platform, since this is poly-T or random and capture, probes. And so we all think they should be able to capture DNA as well. It's just doing for now, our product, the workflow, the kit we provide is focusing on RNA first. But if you want to try something novel, you can purchase our kits and try capture DNA. Okay.
I think we.
Yeah, maybe we should move to Dr. Wang's talk.
Yeah, so
Well-
we will take a break first.
Okay, thank you, everyone.
Yes, thank you so much. So we'll take about a 5-10 minutes break, and you can get coffees. For everyone who is online, please get your own cup of coffee and then come back and join us in 5 to 10 minutes. Thank you. Okay. So we are going to have Dr. Cheng Wang right now from UCSF, who has applied single cell and spatial platforms on the scientific focus of maternal fetal interface. So I'm going to hand it over to him right now to go ahead and introduce us to his research.
Hi, thanks for the introduction. Did you guys see the right screen?
Yes, we do.
Okay. Thanks for the introduction. Hi, everyone. I'm Chen. I'm currently a postdoc researcher from Dr. Jingjing Li's lab from the UCSF. And today I'm happy to be invited here to share with you our experience, our recent experience, where we have tried to combine the use of the cutting-edge spatial transcriptomic technologies as introduced by Yuan today in her talk, the Stereo-seq, with our previous single-cell multi-omic sequencing data to redefine the different cell types at the human maternal-fetal interface in the developing placenta. We know that the placenta is a transitional organ that is only present in the pregnancy, so it is critically important to maintain a healthy development of the fetus. So let me briefly introduce some details and backgrounds about the anatomic structures as well as the developmental timelines for the human placenta. The placenta is the primary regions we're able to identify the maternal-fetal interface, which is a highly specialized regions where the maternal and the fetal tissues come into close contact. And this will allow for the nutrients and the oxygen exchanges to support a healthy pregnancies and the fetal development. And in this interface, the fetal-derived, we can actually observe the fetal-derived structures called the chorionic villi, and these villi were deeply anchored into the maternal tissues called the decidual to create the intervillous spaces for the blood and nutrients supply to the baby. As shown here in this development timeline of the placenta, it compares the development of the fetus over all of these 40 weeks of the gestation. And starting from the weeks of 0, when the implantation happens, the fertilized blastocyst embeds in the maternal uterine wall, and it initiates the development of the fetus and the placenta. And during this time, between the week 0 to the end of the first trimester, which is roughly about the gestational weeks of 12 or 13, actually, in this time, the placenta is in a relatively low oxygen environment. Given that the placenta villi is mostly lined by the fetal-derived cell types called the cytotrophoblasts or the sensation trophoblasts, these fetal-derived cells will start to migrate into the maternal tissues at the end of the first trimester. And these fetal cytotrophoblasts will bridge the maternal uterine wall and begin to reshaping a very specific type of blood vessels called the spiral arteries in the decidual. And this remodeling process will increase the capacity of the maternal blood vessels to make the blood vessel larger, and it will thus provides sufficient nutrients and blood to the fetus. And the placenta will also help to develop to bring more oxygens, and it also functions to remove the harmful waste that are produced by the fetus. Over time, these tiny structures of the placenta villi will develop into increasingly dense branching structures called villus tree to accommodate the increased demands of the developing fetus. So as mentioned in the previous slides, one of the most critical events during the healthy pregnancy is the spiral artery remodulation. And since only these fully remodeled spiral arteries can provide a sufficient blood flow to the developing fetus for its normal growth. When there's no pregnancies, actually these spiral arteries span the tissue of the uterine wall to supply the blood to the surface lining. And in the normal pregnancy, these fetal-derived cytotropoblast cells will migrate out from the placenta villi to invade the intervillous spaces surrounding the spiral artery. And this will end up with the displacement of the endothelial cells that normally lines the blood vessels. And this process is what we call as the spiral artery remodulation. And the remodeled spiral arteries will then help to make the vessel larger and to optimize the blood flow that comes from the mother to the fetus to support its normal function. However, in the normal pregnancies, the fetal-derived cytotropoblast actually cannot sufficiently invade the intervillous spaces, and in some cases, the invasion will not happen within the blood vessels, but instead, the invasion could be deeply, although that the invasion occurs deeply inside the maternal tissues, it's not targeted to the right places within the blood vessels. And in all cases, these insufficient or incomplete remodeled vessels will lead to a very severe conditions called preeclampsia, which is one of the leading causes of the mortality for both mother and the fetus during the pregnancy. So in terms of the cell type composition at the maternal-fetal interface, it is highly complex because we often observe very frequent cell-cell communications between the maternal and the fetal cells in the placenta. Although we have known that there are the functions of the cytotropoblast and the other immune cells from the maternal side could help support the healthy pregnancies, their origins and their exact functions and the mechanisms in supporting the fetal growth has not been fully identified. So in these studies, we're primarily to reconcile this problem by deeply sequencing the different cell types at the maternal-fetal interface. And the most frequent or the most prevalent cell types at the maternal-fetal interface is the fetal cytotropoblast. These cytotropoblast reside within the placenta villi can be differentiated into two distinct subclasses of the subtypes. And for some part of these cytotropoblast, they can be specialized in or fused into the multinucleated cell types called the sensation tropoblast. These cells mostly form the outer membrane of each individual placenta villi, and they are responsible for the nutrients and the oxygen exchanges. Whereas the other part of the cytotropoblast, they can be differentiated into cell types that are highly invasive. And when these cells are migrated out from the placenta villi to invade the maternal tissues, they become as the extravillous tropoblast or the EVTs. And the functions of these cells are mainly to remodel the blood vessels. In addition to the cytotropoblast, we also expect to see different types of the fetal cell types with fetal origins. For example, the half-bowel cells, these are the fetal-derived microphage-like cells, but we don't know yet about their functions in supporting the fetal growth. And also, we are expect to see numerous fetal stroma cells and the perivascular smooth muscle cells that lines the fetal capillary within the placenta villi. As for the maternal size, since the placenta is closely attached to the maternal decidual, we expect to see different subtypes of the decidual stroma cells and a lot of different types of tissue resident immune cells, including the decidual macrophages, uterine natural killer cells, and the T cells. And all of these immune cells can actively responding to the invading EVTs. So a proper recognition between the maternal immune cells and the invading fetal cells could be critical for healthy pregnancies. So in other words, the dysregulation between the cell-cell communication between these two cell types from fetus and the mother could lead to very severe situations such as pregnancy complications, including the miscarriage, preterm labor, or what I have mentioned in the previous slides, preeclampsia. And in this specific study, since we are trying to focus on mostly on the identify the different cell types at the maternal interface, so we particularly targeted to collect the samples at the decidual bacillus, which is near the implantation sites, rather than other type of decidual tissues, such as those tissues from the capsularis or the parietalis. These samples has not been included in our current study because they did not, or very few fetal cells can be identified. So this is a representative overview showing the histology of the human placenta, where at the top, this is the fetal-derived chorionic plate, where the placenta villi is growing from this chorionic plate, and it will develop into the villus trees, and it will gradually invading the maternal tissue in the bottom to form such a maternal-fetal interface, which is the primary regions we're interested in this current study. So first of all, we have confirmed that the tissues we have collected are actually from the decidual bacillus, and this can be achieved by visually checking the presence of the fetal tissues, for example, the placenta villi here under the microscope. And also we have performed the immune staining to co-stain the cytotrophobus marker, the cytokeratin, as well as the decidual stroma cells markers, the vimentin, on the samples we have collected. And here we can see the structure of the both of the floating villi and the ankle villi from the fetal size. And this confirms that the samples we collected is actually near the implantation site. So in order to capture as many as the cell types as we want, we try to include more samples, and these samples go exclusively only from those healthy individuals, which means that they are not with any indicated pregnancy complications. And these samples we have collected has covered the entire gestational weeks or gestation stages, spanning from the gestational week 5 to term. And after extracting the nuclei from these samples, we performed the single nuclei multi-arming sequencing using the droplet-based methods offered by the 10-time genomics. And in total, we have sequenced about 200,000 cells from these microdissected samples from 23 individuals. And for each of the single cell, actually, we have generated the paired transcriptome data by the RNA sequencing and the epigenome data by the ATAC sequencing at the same time for the same cell. We have identified several notable cell types from the maternal-fetal interface, including 3 different subclasses of the cytotrophoblasts and the different subtypes of the tissue-resident immune cells. And we also observed there's a significant dynamic changes in the cell type compositions during the progression of the pregnancies. For example, the fetal cytotropoblast, sensation cytotropoblast, the SCT here shown in the blue color here in the pie chart, it is gradually increased in its proportions over the gestational week. So among these 200,000 cells, actually we have captured about 20 major or more than 20 major cell types from these cells, including 3 major subclasses of the fetal cytotropoblast as previously introduced, the sensation tropoblast or the SCT, the VCT, and the EVTs. We have also captured cell types that shows exclusive maternal origins. For example, the decidual stroma cells within the uterine and the epithelial cells from the endometrium, which are the cell types that usually lines the endometrial glands. We have also detected several cell types with mixed origins, including the endothelial cells and the perivascular smooth muscle cells. And these cells can come from the blood vessels that belongs to the mother or the fetus, but we did not know actually where they can come from. And for these cell types, we actually perform genotype-based phasing to characterize the exact origins for each individual cells. Because we have already generated the paired transcriptome data and the epigenome data for all of these cells, we can project them by using either the test sequencing peaks or the gene expression data. And so basically for each of the cells, we identified with the from the UMAP on the left, we can have their transcriptome data immediately available on the right. So regarding to the cell type annotations, we have annotated each of these major cell type based on the selected canonical markers that have been previously reported in early studies. And in this expression plot, we can see a diagonal trends indicating that the markers we selected are highly specific in 1 or 2 cell types. For example, the TBCT3 is a well-known transcription factor that is uniquely expressed only in the villus cytotropoblast rather than other types of the fetal cells. And the PCAM1 is the gene encodes for the biomarker of the CD31. It is a canonical marker that people have often used to label the endothelial cells. And also these tissue resident immune cells can be characterized based on their specific expression of the immune cell markers. We have also checked the chromatin accessibility data at the promoter regions for these marker genes. And we also observed there's a high consistency between the transcriptome data and the epigenome data. We can see there's a consistent specific cell type specific openness in the promoter regions for this marker in the cell types they used to label. With the provided single nuclear test sequencing data, we can also characterize a set of cell type specific enhancers with each individual cell type. And these enhancers, we have found that they are closely associated with the notable cell type markers. And among all of the cells we have sequenced, actually the fetal derived cytotropoblast account for over half of the total number of the cells. So there are 3 major trophoblast subclasses. The villus cytotrophoblasts are more likely to be a progenitor stage with a very specific expression of the TP63, which is a transcription factor. And these cell types can be differentiated in 2 different paths. The villus cytotrophoblasts can be fused into the sensation trophoblast, which are labeled by the expression of another transcription factor called HOPX. Or these villus cytotrophoblasts can be differentiating into the invasive extravillus trophoblast once they attach to they approach into the maternal deciduals. And these will start to increase the expression of the HLG, which is a very unique marker for the invading EVTs. And from the developmental trajectories, we can also identify a lot of notable terminally differentiated subtypes of EVTs and or the SCTs. For example, for the EVTs, we have identified three major cellular states that are terminally differentiated. The EVTs, or the endovascular EVTs, are referred to those cells that are highly invasive and can be present in the lumen spaces of the blood vessels, whereas the perivascular EVT, the PEVTs, could be those reside near the blood vessels after the displacement of the endothelial cells occurs. The majority of the EVTs actually will be differentiating into the interstitial EVTs or the IEVTs because they are deeply invading the maternal tissues, but the invasion does not correlate with the nearby blood vessels. And for each of these single subtypes of the EVTs, they can be molecularly defined by their marker genes. For example, the endovascular EVTs, they have been known for expression of the NCAM1 and FLT4, whereas the interstitial EVTs, they have increased expression of the AOC1 and the PRG2 once they are deeply migrating to the maternal tissues. In addition to the fetal trophoblasts, we have also a particular interest in studying the maternal decidual stroma cells because they can agnostically regulate the invading EVTs. In our data, we captured 5 major subtypes of the decidual stroma cells, and each of them can be characterized with their own unique marker genes. For instance, the DSC0 represents naive states or a non-decidualized stroma cell state. And their expression was, their identities can be verified by their expression of the ACTA2, whereas the DSC3 and 4 are 2 different subtypes of the decidualized stroma cells. And these cells, these cell types both expresses the decidualization markers, the IJBP1, but they can be distinguished by their unique expression of the semaphorin 3A or SERPINF1. So although we have characterized so many different cell types at the maternal-fetal interface, we actually do not exactly know where these cells can come from, especially in terms of the cell subtypes. So therefore, we seek to employ the cutting-edge spatial transcriptomic technologies to localize them with more precise spatial information. And we chose the Stereo-seq because because these techniques has a lot of advantage for our cases. First, it allows us unbiased whole transcriptome profiling, as Yuan introduced in her talk. Since a lot of marker genes we have used to label the placenta cell types could be non-coding genes, or they have highly variable regions. For example, the HLG, this is a MHC class 2 molecules, and it cannot be efficiently captured using the probe-based method. Besides, the Stereo-seq also help us to reach high spatial resolutions to about 500 nanometers. And this will allow us to reconstruct the transcriptome for each identified single cell in the tissue section after the cell segmentation. And this actually provides us with more accurate information comparing to the deconvolution methods, which is commonly implemented in other existing spatial technologies methods when if these technologies doesn't have such a high resolution. Another advantage of the Stereo-seq is the large capture area, and this is pretty suitable for our tissue type since the prima placenta is comparatively huge if it is compared with other developing tissues such as the fetal heart or fetal brain. And also the Stereo-seq workflow is generally easy to work with because it can start from the capture of the mRNAs using the probes containing the poly-T, as well as there's a UMI barcodes labeling the specific molecules of the mRNA, as well as the CID, which stands for the spatial barcodes that help us to align the location of the mRNA. So basically, after the tissue mRNA is captured by these poly-T probes, we can perform the reverse transcription to capture or collect the cDNA. And these collected cDNAs will be proceed for the amplification and the library constructions, and eventually they can be sequenced on a DNA nanoball sequencer. And here it shows Stereo-seq results from one of our recent run of the second trimester basal plate samples. We have performed the immunofluorescence before proceeding for the mRNA capture and the same tissue section. And based on our immunofluorescence results, we can have a clear overview of the anatomical structure of these particular sections. And from the left, we can see this is clearly shows the structure of the maternal decidual, whereas the majority of the tissues captured in these slides is actually from the fetus, where we can see a lot of floating villi, as well as very few tiny anchron villi that attach to the maternal decidual. And this is the heat map showing that the total number of the UMI counts with the spatial distribution across the different spatial spots. So we employed the data analysis pipeline called SAW that offered by the SDOmics. And this is a integrated pipeline that has included many analytical steps, and it is easy to work with. It includes the steps of sequence realignment, image pre-processing, and the single cell segmentation, as well as a few toolkits for the downstream single cell analysis. So we basically reconstructed the transcriptome for each single cells after the cell segmentations, and these cells can be projected to a UMAP embedding. And by the clustering, we are able to identify the different cell types as we have previously captured from our single cell multi-omic data. And for example, in this expression dot plot, we can see there are the specific expression of a lot of signature marker genes that we have previously used to annotate our cell types in the single cell multi-omic data. And then by projecting these cells back into our tissue slides, we actually can visualize a very clear spatial patterns for the different maternal and fetal cell types. For example, we can see here in the decidual stroma cells that are shown in the green color, they have been spatially lining above the uterine epithelium cells, which is indicated here in the light blue color here. And also at the fetal sites, we observe that there's a clear spatial patterns of the fetal sensation tropoblasts that is shown in the yellow here. They have formed they have the majority of these fetal sensation tropoblasts are identified at the outer membrane of the individual plasma villi. So when it comes to into the each cell subtypes, the Stereo-seq also help us to capture the spatial information for those cell subtypes that we previously do not know about. For example, in our single cell data, we actually observe sensation cytotrophoblast subtypes that highly expresses the genes encoding the secretory hormones, such as the CSH1. This is hormones. And in the Stereo-seq, actually, we noticed that these sensation cytotrophoblast subtypes are not present in all types of the plasma villi. Instead, the secreting sensation cytotrophoblast is only found in few of these villi, and these secreting plasma villi tends to be spatially clustered together. Another example is the ACTA2, because it is a marker that people have often used to label the perivascular smooth muscle cells. But this these positive cells in our single cell data can actually can come from either from the maternal tissues or from the fetus. However, only with the However, only the single cell multi-omic data doesn't offer us any information about which could be the origin of these cell types. But with the spatial transcriptomic data, we actually noticed that the majority of the ACT2 positive cells could be of fetal origins, because we can see the clear spatial patterns that these cells are lines reside within the placenta villi and to support the fetal capillaries. In addition, the Stereo-seq also allow us to capture very refined structures at the maternal-fetal interface. For example, we have captured the anchor villi in these particular samples, as shown here in the box. This is the same anchor villi, and we can see in the bottom of the anchor villi, the IgBP1 is the marker labeling the decidualized stroma cells, whereas the HLG is labeling those migrating fetal cytotrophoblasts out from the cell columns to invading the maternal tissues. So we are lucky that these tissue sections actually capture the moment where the EVTs start to invading this maternal tissues. And the spatial transcriptomic data actually help us to capture that only those decidualized stroma cells rather than those non-decidualized stroma cells can work to antagonize such invasions from the fetus. So here I will try to summarize our current preliminary analysis for our spatial transcriptomic data and the single cell multi-omic sequencing data. So initially we do not know much about the cell types, especially for those subtypes at the maternal-fetal interface. So our best strategy is to sequence as many cells as possible. And this strategy helps us actually discover a lot of different cellular states, and potentially can help us to identify very real or intermediate cell states. However, when we sequence a lot of number of cells, we identify the different subtypes, but actually we do not know where these cells can come from. So we have to switch to our strategies to employ the spatial transcriptomic data technologies to identify the location of these different cellular states at the maternal-fetal interface from the primary tissues. And the Stereo-seq actually offer us an opportunity to identify the precise transcriptome for each of the individual single cells with a very high spatial resolutions. And it is unbiased capture of the mRNAs actually help us to allow us to map our single cell multi-omic data to the spatial to our spatial data to identify the potential location for these real cell types. So this will actually deepens our understanding of the key molecules and their functions in supporting the healthy pregnancy. And thank you for your time. And I would like to acknowledge all the help I have received from our lab member and our collaborator for the access to the tissue. And I will also acknowledge the company Genomics for offering me such an opportunity to share with you our recent experience in employing the Stereo-seq. And thank you for your time. And I will be happy to take any questions you have.
Thank you, Chen Wang. That was really interesting. I'm going to open up to any questions from the audience. We have any questions? Everyone is tired because it's 3:30. No. I just have a small question. So the thing is, it's something that we discuss quite a bit, especially for those of us who are new to spatial. Like when you want to study something complex, like whether it's what do you call it, maternal fetal interface, or whether it's tumor, the spatial context is undeniably important. But the thing is, how do we start? So there's always this question about single cell or spatial. Do you think that you could have come to the same conclusion just jumping straight into a spatial project first? Or do you think that that single cell was actually like quite instrumental or pivotal?
I think both of the technology, these two technologies are actually complementary to each other, because for the single cell data analysis, the single cell did not actually help us to identify the particular location of these cells, but the spatial but it can offer us to have a completely overview of what could be the cell types that present in the tissues you have. So it is an unbiased capture for the different the different cell types. But for the spatial transcriptomic, personally, I believe that this is the strategies we're going to specifically to capture the area of interest. So because sometimes the sections, the variation between the different sections could be huge. So you have to visually check under the microscope to determine what would be the structures you're going to be interested in, and then can proceed with the spatial transcriptomics to verify the findings from the single cell analysis.
Thank you. Does anyone have any questions? Okay, so no questions at all? All right. So I just really want to thank both our speakers. One of the things that I really want to point out is that this talk was really good in a sense that it's really not about choosing spatial or single cell, but it's about combining all of these technologies to answer your question. And we hope that everyone who attended actually had that message passed across. Yeah, because I think the platforms, we don't do the platforms any justice if we do not understand how to use it and also when to use it. So that was, thank you again to all our audience for coming, and also for both our speakers for attending and for giving such fantastic talks. So with that, if there are no more questions, I would actually like to end the session. And please feel free to email us or to reach out to us if you have anything else. So yeah, thank you. And yeah.
Thank you.